To begin, we will start with interphase. During late interphase, the nucleus is well defined and surrounded by the nuclear envelope. It contains one or more nuclei. Just outside the nucleus are two centromes, formed during early interphase by replication of a single centrome. Microtubules extend from the centromes in a radius array called asters. The chromosomes have already been duplicated but are still in the form of loosely packed chromatin fibers. During prophase, the nuclei in the nucleus disappear. The chromatin fibers become tightly coiled and condense into chromosomes. Chromosomes line up in pairs and are joined at the centrome. In the cytoplasm, the spindle fibers begin to form and are made of microtubules. The centromes move away from each other, propelled by the lengthening microtubules or spindle fibers between them. In metaphase, the centromes are at the opposite ends of the cell poles. The chromosomes are lined up at the metaphase plate, an imaginary line equidistant between the two poles. The centromeres of the chromosomes are lined with one another. The chromatids of each chromosomes are attached to the microtubule which form the spindle. In anaphase, the paired chromosomes separate. They move along the microtubes toward opposite poles of the cell. The poles move farther apart. By the end of anaphase, the two poles of the cell each have a complete set of chromosomes. In telophase, the microtubules becomes even longer, and daughter nuclei begin to form at the two poles of the cell. Nuclear envelopes are formed, the nuclei reappears, the chromatin of the chromosomes uncoil, mitosis is now complete. One nucleus has divided into two genetically identical nuclei. Cytokinesis follows and involves the formation of a cleavage furrow which pinches the cell into two.